In this video, I'd like to go through the last part of the notes for Unit 5. So we left off talking about action potentials, and I want to just review the action potential quickly before getting into the new material. And so we covered the action potential, so bottom of page 6 going into page 7, and I'm going to jump over to a picture. And so what an action potential is an electrical event that is only generated by an axon. So the action potential is what people call a nerve impulse. It's generated by the axon, and once it's generated by the axon, it will be conducted down the axon full strength, right down all of the teledendria, all those little branches, and sweep across all the axon terminals that neuron has. So the action potential, like I said, it's special in that it it is a nerve impulse. Okay, and it's only generated by the axon. And so, summary to how the action potential works. This is a picture from our book. Is that, remember, the neuron is spending a lot of energy to keep its interior at -70 millivolts compared to the outside of the cell, and so that's known as the resting membrane potential. So here what we're doing is we're looking at the axon specifically. We're not looking at the dendrites or how it receives incoming information. We can try to put that together in the end. But the axon is resting at -70 millivolts. And if it is depolarized to what we would call threshold, or threshold being just enough of a voltage difference, enough of a voltage change to open the voltage regulated sodium channels that are only located in the axon and the axon terminals and downstream from the axon. So that's why only the axon can generate an action potential. So here we can see a depolarization occurring. Our textbook is using -55 millivolts as threshold. In my notes, I'm using -60 millivolts as threshold, and this will vary cell to cell. So we're ballpark talking about the same thing. Okay, there's no absolute number that we're gonna go by. So on the test, for instance, I would tell you what threshold is. I'm not gonna expect that you know that. But so -60, -55, I don't mean any difference in the notes. So that depolarization took the cell 15 millivolts more positive because it's resting at -70, so now it's at -55. That's the trigger for voltage regulated sodium channels to open. Sodium enters the cell, and because a positive charge is entering the cell, you can see that inside of the axon is becoming more positive. It goes up to about positive 30 millivolts. And right around this time, what will happen is the voltage regulated sodium channels will inactivate. They don't technically close, but they don't work anymore. So they'll inactivate, and the voltage regulated potassium channels that are in the axon will open. And when the voltage regulated potassium channels open, potassium leaves. And so that's the repolarization. So here potassium is leaving. And those potassium channels, just like they're slower to open, they're slower to close too. And because they're slower to close, more potassium keeps leaving, and so much positive charge leaves that we actually go below resting membrane potential, what's known as a hyperpolarization. And that's due to this, long, lingering, open potassium channel, and potassium just keeps leaving. But as soon as that potassium, the voltage-regulated potassium channels close, the sodium-potassium pump is going to be able to use active transport to reestablish that stable difference that we've learned about, the resting membrane potential. So this is what is known as an action potential. This picture in our book, I think it's really nice. It's useful. So it shows you the different parts of the action potential as far as the voltage regulated sodium channels and the states that can be in, closed, inactive, or closed, open, or inactivated. And then the voltage regulated potassium channels, same thing, what it looks like closed, what it looks like open. And then in our book, they also show you the status of the channels at each different phase of the action potential. So I hope this is helpful too. So we know that you have to get the voltage regulated sodium channels to open. If you can do that, sodium is going to come in to the cell, causing that depolarization. Next, what will happen is the sodium channel inactivates. potassium channel open, so sodium can't cross anymore. Potassium's open, so potassium will leave. That leads to our repolarization, and then the hyperpolarization is because that channel is staying open longer, and the sodium channel will close. So that is a little review of the action potential. And one last thing about the action potential before I technically get into, our brand new material today, because I think this is a hard thing to understand. So we look at this graph and we analyze this graph and we know what's happening at every point on the graph, but really what does the graph represent? And so this is a picture from our book where they're showing us the recording electrode. Okay, so this is a glass microelectrode that's inserted into the axon. So this is what's, doing the reading. And so do you see how we're looking at this point in the axon over time? And so at this point in time, even though they're showing me in red, an action potential is going to come up to this point. Do you see how on a graph, if I'm recording at this area, we're at resting membrane potential? So what that graph represents is an action potential sweeping across that spot where we're recording. So that's what they're showing us here, that when the action potential sweeps across that electrode, that's when we see this characteristic graph. I hope that makes sense. And the action potential will keep moving forward. And so, for instance, even though at this point we're seeing the hyperpolarization, and that's that hyperpolarization that follows the action potential, that's going to assure the action potential is going to move in one direction down towards the axon terminals and keep propelling itself, towards the end of the cell. So I hope that makes sense too. That's a hard thing to understand and to explain. So when we look at this graph, we're literally just looking at this one tiny pinpoint of location at this one tiny millisecond of time. And we're just seeing that, there was a voltage change that occurred in that millisecond at that point. Okay, so that's an action potential. So moving on to new material for today. In the notes, what I start to talk about after the action potential, a lot on the action potential. Okay, so top of page 10 is what's known as a refractory period. And so a refractory period just means that there is a period of time when the neuron doesn't work. In other words, it can't generate a continuous action potential. So to contrast this to muscle, if this made sense to you in muscle, if we stimulate the skeletal muscle fast enough, what we can do is we can get a contraction and it stays contracted over time. So you might remember that the wave summation or the incomplete or complete tetanus, right? So we give, one message after another, frequent, stimulus, and the muscle contracts and it stays contracted over time. So with a neuron can't do that. And I'm just going to jump to a picture here from our book. So in this picture, what they're doing is they're stimulating an axon just artificially. So we're not looking at like what's happening in the body, just, we're giving it a voltage just like we did the muscle to see what happens. So of course, if we hit it with a sub-threshold stimulus, neuron stays at resting membrane potential, or axon does, I should say. That makes sense to us, right? And so if we give it a threshold stimulus, here I can see for the entire time that we're delivering a threshold stimulus to the axon, that the axon is generating action potentials. So in this experiment, what we can do is we'll give it a little break, and now I'm going to give it a stronger stimulus. What would happen if we stimulate the axon even more? And you can see that action potentials are generated even quicker. And so there's no such thing as, what we saw with skeletal muscle in terms of like tetanus or anything like that, where you can have an action potential and like keep having an action potential. So you see how action potentials are individual events. And what you can do with stronger stimuli is you can make the axon generate action potentials faster and faster. But they work as individual events, and you can only, with a maximally stimulated axon, you're only going to be able to get action potentials being generated so fast, which is very, very fast. But still, they're going to be individual events. And notice how the action potential is always the same. So remember that graph topped out at the positive 30 millivolts, and I noticed that there's no such thing as a bigger action potential. It's all or nothing. So what happens when you have stronger stimuli is that you get more action potentials in a period of time. So I hope that makes sense. And there's a limit to this. And let me give you an explanation for this. So the reason for that is once we open the voltage regulated sodium channels and initiate an action potential, sodium enters the cell, causing that depolarization. At this point, the sodium channels inactivate, right? And the sodium channels don't start to close until, we're around like the threshold resting membrane potential. That's when the sodium channels are going to start to close, which means that, and if you drop a line down, which means that the cell has no way of responding to another stimulus at this point in time. Because remember, if you want to initiate an action potential on the axon, what we have to do is depolarize it to threshold. What's the result? The result is it opens voltage-gated sodium channels. So in other words, the neuron's unable to respond to a second message until the voltage-regulated sodium channels are closed. And so inactivated doesn't count. So once the voltage regulated sodium channels close, now we could respond again. But for this entire period of time during that depolarization and repolarization, this is what we call the absolute refractory period. Because even if you did receive a strong stimulus during this period of time, what more is there to do? Because the voltage regulated sodium channel is already at work. Okay, so that's the absolute refractory period. The relative refractory period means that the voltage-regulated sodium channels are closed. Okay, so we could generate another action potential. We could stimulate the axon enough to open those voltage-regulated sodium channels again. But here's the thing. At this point in time, remember how there's a hyperpolarization that follows an action potential? It makes it harder to reach threshold. So the threshold is still the same. So if we're saying it's negative 60 millivolts or negative 55 millivolts, wherever we're placing that threshold, that doesn't change. It's just that when you're in a period of hyperpolarization, you're further away from the threshold. So previously, like in this picture, a 10 millivolt change would get the voltage regulated sodium channels to open. If I'm in a hyperpolarization, I might need a 20 millivolt change. Do you see the difference there? So you can still elicit another action potential. It's just harder during the relative refractory period because of that hyperpolarization. We're further away from threshold. And so because of these refractory periods, this is an explanation for why an axon is going to have to generate individual action potentials. It's never going to fuse into, one sustained action potential. So hopping back to the notes, I talk about that here, and then I also define, what is the absolute refractory period, which is when basically the voltage regulated sodium channels, they're already working. They're either open or inactive. Whereas the relative refractory period, that's during the hyperpolarization. So your sodium channels are closed, and you can trigger another action potential. It's just harder, right? Because we're further away from threshold. So that is an explanation of the refractory period. So as far as, how quick is the action potential going to go down the axon? The conduction velocity, which is the speed at which the action potential will travel down the axon, has two, primary factors that influence it. One is the size of the axon, the diameter, and basically the bigger the axon, the faster it's going to be. And so these axons, if you want to think of it as, large, medium, and small, they're ranked in type A, type B, type C. And so I have that defined here, but basically the bigger the axon, the faster that message is going to travel. And then the other big factor is the axon myelinated? If it is myelinated, myelination increases the conduction velocity. So I gave some pictures of this, so let's see if this makes sense. So conduction down myelinated axon, that's known as saltatory conduction. And the way that this works is if we generate an action potential at the axon, so here we can see there's an action potential because the inside of the cell is at positive 30 millivolts compared to the outside. Sodium has entered the cell, and this is a myelinated axon. And so what happens is that sodium will be enough to change the electric charge where it's not myelinated, the next patch that is unmyelinated, which is known as the node of Ranvier. So it's going to be able to depolarize the node of Ranvier to threshold. So here you can see it's coming to -60. That's threshold. And so what that's going to do, it's going to trigger an action potential. It's going to open the voltage regulated sodium channels. Sodium will enter the cell. The cell will become depolarized and even become more positive, positive 30 millivolts. And that sodium is going to go through, just think of this as like a speed tunnel, and it will definitely be enough to change the electric charge at the next node of Ranvier to threshold, and so on. So that's conduction on a myelinated axon. Basically, what people describe it as is the action potential is generated in the nodes of Ranvier, and so it jumps down the axon, and so it's quicker. You can imagine if we were in like a single file line to the movie, and there's 100 people in this orderly single file line, and person at the counter hears that the movie sold out, and if you yelled, sold out, and the next 20 people heard, and then, person number 1920 also yells it sold out, then that message jumps down the line. That's like saltatory conduction. That's compared to an unmyelinated axon. Unmyelinated axon, that's known as continuous conduction. And this is a picture of continuous conduction. It's basically the same thing. It's just that you don't have like the quote unquote like speed tunnel, if that made sense, that will take the depolarization, the sodium, to the next node of Ranvier. So if we have the initial patch of axon experiencing an action potential, voltage regulated sodium channels come in, this is going to positive 30 millivolts. Remember, threshold is only, in our example, -60 millivolts. So if this little patch of memory is going to positive 30 millivolts, there's no question that there is enough voltage change here that the next patch will be brought to threshold, right? And when you bring that patch to threshold, that opens the voltage regulated sodium channels. Sodium comes in, and I know that's going to be enough of a change that you're automatically going to trigger the next patch of voltage regulated sodium channels to threshold, and so on. So that's how continuous conduction works. With continuous conduction, you can't skip any areas on the axon. It's just going to have to move down the axon step by step by step. Well, to use my movie analogy, this would be like, in the 100-person line that person number 1 says to person number 2 to 3 to 4, and you can't skip anybody. Everybody has to hear this message. It's going to take longer. Another example, analogy could be, say, like if I wanted to leave the room. Maybe I could leave the room like in 2 leaps, or if I had to cover every little piece of the floor, does that make sense? Then maybe it would take me 20 steps. It's just going to take me longer. So that's the difference between saltatory and continuous conduction down the axon. Now, once that action potential sweeps across the axon terminals, that's going to trigger release of neurotransmitter into the synaptic cleft. And this is territory that I think we're more familiar with from the neuromuscular junction. So see what you think. So here, when I'm talking about synapses, notice that I'm talking about 2 types of synapses. The chemical synapse, this is really a review of what we've already talked about because the neuromuscular junction is a chemical synapse. I'm going to review that with you. The other type of synapse is called an electrical synapse, and that's when there are gap junctions that connect the cells. And so in other words, if there is an ion change in one cell, those ions will actually cross these channels into the neighboring cell, and so you don't need neurotransmitter. It's literally like they function as one cell, kind of. And we'll talk about this in AP2 when we talk about how the heart operates, because the cardiac muscle is going to operate via electrical synapse. But chemical synapse, this is exactly like the neuromuscular junction. I'm going to switch over to a picture from the book, see if this makes sense. So here's a picture from the book. So they're showing us in the pink lines, if the action potential sweeps across the axon terminal, that's going to trigger the opening of voltage regulated calcium channels, and that calcium is involved in the exocytosis mechanism. That's all. So calcium enters the axon terminal, and that's going to trigger some of these synaptic vesicles to release the neurotransmitter into the synaptic cleft by exocytosis. Now, if we're talking about a chemical synapse in general, the neurotransmitter that could be released could be acetylcholine, or it could be something different. It could be epinephrine, norepinephrine, dopamine, serotonin, right? So lots of possibilities here, and we're not worried about what it is, to be honest. We just want to know that the neurotransmitter, the generic neurotransmitter, is released into the synaptic cleft. And when it's released into the synaptic cleft, it's going to bind to receptors. And then I increase the size of this part of the picture for us. So when the neurotransmitter binds to the receptor, do you see how they're just making this generic? This is going to be a chemically gated ion channel. That chemically gated ion channel is typically closed, but when the neurotransmitter binds, it opens. Here they're showing us, like the generic arrows. All that will happen is passive movement of the ions that are allowed to or are able to pass through this channel. And so, do you see how we're back to the beginning of the story here? I hope you do, because this is a graded potential. So let's say, for instance, this neurotransmitter opened sodium channel. Then I know sodium will enter into the cell. That's going to be a depolarization. That depolarization is going to start to spread from that area. That's what's known as a graded potential. It's also possible that the neurotransmitter bound to a chemically gated potassium channel. If a potassium channel opens, what will happen is potassium will leave. And when a positive charge like potassium leaves, it makes the inside of the cell more negative. That increased polarization or hyperpolarization, the increased negativity inside of the cell, that will also spread. And that has an inhibitory effect. And so it's possible that this graded potential is either excitatory, it's a depolarization, it could lead to an action potential, or it's inhibitory, that it's going to bring the cell further away from the threshold level by being in a hyperpolarized state. But in either case, what will happen is that neurotransmitter will have a limited, lifespan, because it's either going to get enzymatically degraded, for example, acetylcholinesterase, or it could just diffuse away, or sometimes it's recycled by the presynaptic cell, by the axon terminal. And so all of those different things to get rid of the neurotransmitter are possibilities, all those different explanations. So just to go back to the notes, I wrote this out, how the chemical synapse works. So there's my written explanation of what I just described. And moving forward, this is what I was just talking about. Basically, when that neurotransmitter is released into the synaptic cleft, neurotransmitter binds to the next cell in line. We would call that second cell in line the post-synaptic cell. I hope that makes sense. So it's just, see if this makes sense. I'm going to draw on this and then I'll erase it. So if I had one neuron and its synapses with a second neuron, okay, we'll say synapses with the second neuron. So neuron number one, I would call that the presynaptic cell, right? So here's the presynaptic cell. And then we'd have the synapse, the synaptic cleft in between the two. And then that second cell in line, I call this the postsynaptic cell. And so do you see when you use this terminology, presynaptic and postsynaptic, what you're doing is you're, talking about a synapse in between the two. Okay, because it's not like this is absolutely the postsynaptic. For instance, if this blue cell released neurotransmitter onto a third cell, and I was talking about the release of neurotransmitter, I'd call this the presynaptic cell. And so what we're talking about when we talk about the postsynaptic cell is the cell that's receiving the incoming information, right? That's the postsynaptic cell. And whatever that information is, remember we said it's a graded potential, it's also known as a postsynaptic potential. So a graded potential and a postsynaptic potential, they're synonyms, okay? It means the same thing. Here, I'm just giving some terminology to opening the sodium channel or opening the potassium channel. So I'm going to get rid of my markup here. Okay, so if the neurotransmitter opens the sodium channels, we know that's going to lead to a depolarization. That's an excitatory event, right? Because that depolarization could drift towards the axon, and that could open the voltage regulated sodium channels. So we call that an excitatory postsynaptic potential. If it opens chemically gated potassium channels, that will cause a hyperpolarization. That could also spread to the axon hillock, but it's going to spread a hyperpolarization, making it further away from threshold. And so that has an inhibitory effect, and we call that an inhibitory postsynaptic potential. Okay, so here we're just naming the type of effect. It's still a graded potential. We're literally back at the beginning of this note packet, okay, talking about the exact same thing. So to wrap this up, some of the dynamics, there is a short lag time that it takes for one cell to communicate with the presynaptic cell to communicate with the postsynaptic cell. That's known as synaptic delay. You know, it's a fraction of a millisecond. It's very short, but if we were looking at, you know, like a pathway with one synapse, that would be faster than, say, a pathway with 2 or 3 synapses. That's all. And then as far as fatigue goes, with synaptic fatigue, neurons don't run out of ATP. Neurons are insulin-independent cells. They can use glucose whenever they need it, basically. And so in this case, fatigue is if we use up all the neurotransmitter. Okay, so synaptic fatigue is neurotransmitter supplies being depleted. And so the last concept that I have in the notes to consider is summation. And summation is just consideration of the fact that it's not going to be this simple, you know, too bad I erased the picture, the presynaptic neuron, synapse, postsynaptic neuron. Let's look at a picture from the book. It's going to be more like this, where the neuron has multiple incoming messages, and then the neuron is going to have to somehow put all those messages together. And just like a math problem, you know, to put all the numbers together, to add it all up, we'd call that a sum. And the same thing with the neuron. It's receiving all these multiple messages, and somehow it's going to have to put that all together, and that's known as summation. So with summation, there's 2 main types of summation. In this picture, and I would look at this from your book, they are reviewing the excitatory postsynaptic potential and the inhibitory postsynaptic potential. So that's worth looking at. But I'm going to look at this picture from our book, which shows, talks about a little bit about summation. So with summation, a few different ways neurons can pay attention to what's going on and how cells that are interacting with the neuron can have a different effect. So here we're just seeing there's a subthreshold stimulus. Here's a second subthreshold stimulus. Okay, so that's no summation. That's, you know, nothing different than what we've talked about. But here, temporal summation. Temporal means time. So if we deliver, if the neuron receives, let's say, if the neuron receives 2 excitatory postsynaptic potentials closer together in time, so do you see how the green arrows are far apart here? Here we're moving those green arrows closer together, that it's possible when you get that depolarization that you can piggyback another depolarization and that is going to reach threshold. Okay, that's what's known as temporal summation, or it's adding up things over a short, you know, succession of time. An analogy or metaphor that I have for temporal summation, if this makes sense, if instead of initiating an action potential, if our goal was to fill a bathtub, you can't fill it with one bucket, but if I empty that bucket into the bathtub over and over and over and over again, that could end up to be a full bathtub. Sorry about that. I had to take a call from my sister. She's in Mexico, so I had to take it. But temporal summation is, like we were saying, if we receive 2 stimuli, 2 like subthreshold stimuli close together, that could add up to a threshold stimulus. And now spatial summation is when the neuron is receiving information from 2 different spaces. And so with temporal summation, this one neuron is saying over and over and over again in rapid succession, you know, say a subthreshold stimulus that adds up to threshold. Here we're saying that 2 neurons are synapsing, or 3 or 300 neurons are synapsing, and they all synapse at the same time. And what would happen is those would all summate. And so two subthreshold stimuli coming from a different neuron at the same time, so different location, that's the spatial summation, but at the same time, time wasn't a variance, then that could also summate to a threshold stimulus. And along with this idea is, say, like if you received an equal and opposite, like an excitatory and a an excitatory and an inhibitory postsynaptic potential of the same magnitude, they could cancel each other out. And so just to take a look at this is from a different book, but it just shows really nicely, the excitatory postsynaptic potential is a depolarization. They let that go to resting. And here you can see time 2, they're going to hyperpolarize the axon, so that's an inhibitory postsynaptic potential. And then at time 3, what they're going to do is they're going to deliver an equal and opposite excitatory and inhibitory postsynaptic potential. And just like a math equation, if I have a positive 10 and a negative 10, they cancel each other out, and you can just see the flat line. So that is spatial summation. So going back to the notes, I described that here, and that is it. So let me know what questions you have, and have a nice week. Thank you.